All right. Hello, everybody. Welcome back to ESM 105. I hope you all had a great weekend. Thank you for your well thought out discussion participation. I actually have started responding to a lot of those because I like what y'all are saying, and I want you to know that I am actually reading them and not just going 3, 3, 3, 3, 3, 3. So I really appreciate your participation. It's great, and I'm really happy to hear about some of the things people are enjoying about the course. All right. So you did quiz 1. Overall, everybody did pretty darn well. Okay. If you want to check your quiz responses out or discuss anything with me, I got office hours. Here they are. Office hours. Mondays 12 to 1. Tuesdays and Thursdays 10 to 10:50 in AMH 169. It's the curved building over there. All right. We are in week 4, so we're finally today going to start our week 3 through week 4 PowerPoint. This weekend, I finished revising the week 5 through week 6 PowerPoint, and I realized that I was kind of front-loading things too much. Based on our syllabus, it would make sense for a few of the topics we're talking about in weeks 1 through 2 to be in week 5 through 6. So it means that weeks 5 through 6, the PowerPoint's going to be easy to get through. All right? And I'll be doing a lot of, remember, we spoke about this in week 3. Here's a reminder of that. So we're on track, no need to worry. You've got a quiz coming up eventually. I think it's in week 5. In week 5, September 28th is your next quiz. That quiz is going to cover material from the week 3 to 4 lecture PowerPoint, as well as the future of life. All right? So same sort of thing. You write me a little brief, it's a very brief essay thing about the future of life, so I can tell that you read it, and you answer some multiple choice questions about the lecture material. Cool? That's not this week. It is next week by the 28th of September. Questions? Okay. Each week, check the week, you know. Yeah, this week's is just keep reading the future of life. And don't forget about those discussions. They're every week. Okay. Any overall course questions, anything else before we go ahead and get back into the material? I'm stoked to wrap up. We're finally going to get into some very relevant topics about food chains, food webs, and carbon, nitrogen, phosphorus cycle. All right? Super relevant topics for you as conservation biologists or whatever field you decide to go into. That's, ecology adjacent or is just ecology, okay? And we really needed to lay out a lot of we really had to do a lot of groundwork to get to the point where we can discuss this stuff, okay? Okay, good. Energy moves through ecosystems, right? We've talked about these state variables and how energy moves through systems, okay? We've got that primary productivity where the initial high-energy organic molecules get generated, all right? I talked to you about how there's different types of primary productivity. A few people really seem to like the chemosynthesis or the chemoautotrophy discussion. So I put a little slide about that in week 5, so we'll talk more about it there. It's really cool stuff, okay? Also, we defined populations and communities. It's also really nice to see how many people engaged with the concept of the species definition. It's really important in the sciences that we have well-defined terms, okay? So understanding what a population is, what a biological community is, essential. All right? Cool. This is the biological community, it's the living piece. We're going to keep moving forward. Food chains, food webs, they demonstrate how energy flows through ecosystems. Usually it's taught as what eats what, right? So I believe, introduced primary productivity to you. That was where we kind of ended last week. Is everybody comfortable with that term of primary productivity? Very good. Okay. Oh, and productivity here is defined as biomass production. Okay. Good. Good. What's the ecosystem? We talked about it. Here we go. Defining the boundaries of a system is difficult. It's the whole system thing that I've been talking about since, I think, day 2. You know, a system, it's all about setting up the boundaries. Where do we stop considering the system, okay? Or you look at a system, right? You look at your, say, your biome or your specific forest or the organism, and you can talk about what goes into that system and what comes out of that system, but you're studying what's going on inside of that system. So you set up your boundaries and you focus your study on what's going on within those boundaries. And that's your system of interest. So right here, we've got a classic sort of food pyramid trophic structure discussion. Okay, let's go into it. You've got primary producers at the bottom, all right? There will be the most biomass of primary producers in any ecosystem, okay? Because every time there's another level put on top of it, there's going to be inefficiencies as they acquire the food, eat the food, and convert it into biomass. Even if you want to talk about it from fundamental physical principles, the laws of thermodynamics dictate that the energy held in this biomass, as it's transformed into this biomass one level above, that's an energy transformation. Some energy will be lost as heat, and it will just radiate out of the system, okay? So we get from this something called like the 1/10 rule, whereas you go up through trophic levels, it'll be 1/10, all right, the amount of energy held in each trophic level. You see those numbers right here on the right side. It's all due to inefficiencies in acquisition of food from the lower trophic levels, and then inefficiencies in converting the energy from one form of biomass into another form of biomass. Is this a concept that you've all been introduced to? Or does anybody need would anybody like additional detail about this portion where I talk about the different tiers and how energy is lost as you move from level to level? Please, yes, and then I'll get yours.
Animals eat other animals? Is it like they like lose energy, I guess?
Yeah.
In their bodies or something? Their bodies are warm.
Their bodies are warm, yeah. So they're losing energy there in carrying out their metabolic reactions. Like just existing takes maintenance energy, right? You're breaking down compounds, and when you break down, like let's say you eat a delicious a BLT, and it's really good. You don't immediately accrue the BLT's worth of biomass, right? So there's all this stuff, all this energy lost in the conversion of it to different forms, and then you're maintaining things like, you know, your heartbeat and all of the processes that are involved in maintaining homeostasis. Driving cellular respiration is energetic, loses energy, right? Cellular respiration is a form of energy transformation. So there's all these different explanations for why you can't just convert every bit of energy in the biomass of something you eat immediately into biomass, all right? But at the very basic level, it comes down to that second law of thermodynamics, where as energy changes forms, you lose energy due to heat to the environment, and it's not sort of captured in the system again. Solid? I love it. Yes, please.
Would the decomposers be considered like feeding back into the like bottom tier, or because it's only considering biomass, it doesn't really count?
So the decomposers are usually considered as sort of their own thing here, right? That interact with all levels of the trophic structure, because the decomposers receive inputs from every single trophic level, because everything perishes and everything then decomposes, right? Now, things consume the decomposers sometimes, and things consume the breakdown products of the decomposers. So they're more complicated than that, right? That make sense? Solid. Yes?
Can you think of an example of a decomposer that consumes light energy?
I mean, we could talk. Most lichens are not designed to carry out that much decomposition anymore, but that'd be like a symbiotic relationship. Decomposition should not be considered as an autotrophic process. Decomposition should be considered as a heterotrophic process for the purposes of understanding the energy flow through ecosystems. Yeah. Nothing else is springing to mind. Yeah, which is really cool. It's a super important type of like primary. It's a type of primary productivity for like a mineral input into the system or generating of a physical matrix in the system, which is really, really important, right? To convert rock into smaller particles, which can then foster growth. It's all, yeah. But as far as talking about energy flow through ecosystems, which is what this is meant to illustrate, the decomposers sort of sit to the side of this and take inputs from every level. Decomposition, remember, this will be very important. Decomposition is cellular respiration. So it takes high energy organic molecules and oxygen, and it produces CO2. So for example, when you disturb a soil that's been stable for a long time and is holding a lot of carbon in it, you introduce all of this oxygen, which allows cellular respiration to run rampant, which generates all of this carbon dioxide and other greenhouse gases. So disturbing and aerating a soil increases decomposition rates, which affects carbon cycling. It's all very important, and that's because the decomposers are carrying out respiration, right? Okay. Systems, primary, secondary, tertiary, quaternary. It would be really convenient if this is how ecosystems actually worked, but of course it's not. Instead, you've got all of these complicated food webs, right? Where the snake eats the bird and the frog and also the mouse, so it feeds on different levels below it, all right? And sometimes so you can classify certain organisms as at multiple levels of the trophic structure, where sometimes they're acting as tertiary consumers and sometimes they're acting as primary consumers, like in an omnivore, right? Fair enough? Solid. So we use food webs, something which, I'm pretty confident that we've seen before. Here's an example of the food web. Okay, let's get the slide shown. I like to have a lot of extra text on these so you can use the PowerPoint as a good, as a review source, all right? But I'm gonna try to break my habit of making sure that I hit every single thing here, I'm just gonna go with what is key in the lecture. But if you have questions, just slow me down, you ask them. I think I hit everything on here. Cool? Awesome, I love it. Okay, so here's a food web in the soil, all right? So look, you see how many things are interconnected. I chose the soil food web instead of doing one of the standard like terrestrial food webs because it's just, I don't know, I wanted it to be a little more fresh and interesting, hopefully. So check this out, right? You've got these plants, the primary producers, they generate organic matter when they die and decompose. So now you've got the decomposers inside of the food web, right? Generates these bacteria. You have fungi and bacteria consuming the organic matter. These are consumed by nematodes, arthropods, protozoans, all right? And then you got larger nematodes that eat these nematodes, but also eat the protozoans. And then these arthropods eat the nematodes, and these arthropods eat different nematodes. And these birds eat these arthropods, and these animals eat these arthropods, and then other animals eat these animals, all right? So here's just a cool soil food web, a little more small scale, just to think about. The life in the soil is incredibly important, and it's overlooked, and it's understudied, and it's still, if you want to do research, they need people willing to do soils research still. It's one of the least well understood aspects of plant morphology and plant physiology and ecophysiology is how rooting dynamics actually change in response to environmental stress. Because guess what people don't want to do after spending 12 years in higher education? They don't want to dig in the dirt and then lay out. This is the job. I used to have to do this in graduate school. You core the soil, you take the plant, you lay it out on a table, and you spray it with water, and you have forceps, and you pull out the rocks, and you spray out the water, and you do that for like 4 hours, because it all will start to rot immediately. Then you take it to a scanner, and you scan it, and then a piece of software processes that and tells you about the roots and how many branching points they have, and then you publish that. It's a great sort of vacant niche in research. No one wants to work in soils because it's difficult, you know? Oh, it's horrible. You just have to do it again. Yeah, and then you have to grow the plants again, or you have to hope that you had backups. So we'd always have backups. Because I definitely got overwhelmed and let all my roots rot in the fridge before, destroying, 2 months of work. It's great. It's great. It just takes me back. This is a way better job. This job's so much better. Sometimes it's hard, to remember that. All right. Food webs, okay? We always introduce them in ecology, environmental science, conservation classes as like, look, it illustrates the interconnectedness of the different organisms alive in the environment, blah, 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 right? But I want you to know, without worrying about all of these details, because they'll come back again later, that whenever you talk about something like this in the abstract, there is a quantifiable way to represent it. And there are metrics about food webs that we can present, okay? Things like connectance, okay, which tells you how connected the food web is, all right? Linkage density, all right, which also tells you how linked up the food web is. Chain lengths, compartmentization, all right? Like, is there a certain portion of the food web where all of the energy sort of cycles around, but there's one input into it? Like, these are not things you have to memorize, but I really, really don't want you to think that if you were to publish something where you evaluated a food web, you would just show a picture like this. There are lots of metrics that people who study these things use to describe food webs. And that is what that slide is for. Yes?
Do these metrics measure how good the food web is?
That's a fantastic question, and one that is very up for debate, right? So just like people have the generalization that a community with higher diversity is more resilient and is more stable. And that is not always the case. Sometimes low diversity communities are hyper resilient and very stable, and the food webs there can be super connected. So this could be a contributing factor, all right? But it's not that every community that has high connectance of the food web is going to be more stable than a community with low connectance. Do you see what I mean? Yeah, so it doesn't necessarily measure better or worse, but it tells us a specific aspect of the food web. Okay. Yeah. And I am very aware that I say web strangely, so I hope it's not too distracting. Yeah, every time it comes out of my mouth, I'm like, should I make an announcement about this? Like, it's okay? Washington. Yeah, explains it all. We say milk, we say wib, we say pen, we say dragon. Yeah, not all of us, but I do. All right. So, cool. What are we doing here? We're doing the trophic levels. We've talked about this. You know these terms already, except for maybe detritivore. All right. Detritivores consume like plant debris. Detritus, exactly. Like dead organic matter that sits on top of the soil and hasn't been converted into into humus yet. Yeah, please.
Why are they in a different class from decomposers?
Absolutely, because the detritivores specifically, they're also called like the shredders sometimes. They work to initiate the first stage of breakdown of organic matter. First, you break it into small pieces, and then the decomposers decompose into, say, constituent macromolecules.
Like bark beetles and stuff like that?
Yeah, things that chew, things that shred. Yep, larger arthropods. Yes.
So I know vultures, they would be scavengers, right? And so the vultures would eat the carcass.
Yeah.
And the detritivores would get what's left of the carcass?
That's a great way to think about it. Absolutely. Yeah. Okay. Usually when we're talking about scavengers, we talk about animals, and when we're talking about detritivores, we're talking about plant material. But I really love the framework for thinking about it, so I'm going to say, yeah, thumbs up. I see another question. Okay, I'm stoked. This is going well. The levels, just ask questions if you got them. We gotta get into, yeah, I've talked about all this. I've got too many slides about this energy flow stuff. I just couldn't help myself. Energy's always lost in the food webs as you go from level to level. Okay, look, this sort of quantifies. Some gets digested, some's not digested, some's used for respiration. All these conversions lead to losses in heat. Then you get some actual body growth, and so this leads to that 1/10 rule, where like 10% of the biomass at the lower level is actually stored as biomass in the next level. Okay, and it keeps getting smaller and smaller. That's why we usually don't have consumers beyond the quaternary level. Question?
So is it like a curved graph of energy loss as you go up the food chain, like each level kind of
Yeah, yes, it's exponentially less each time because you lose 10% or 90% between each level. So it goes like, doop, right? That's the sound it makes. Yeah, boop. It's pretty good, right? I know all the graph sounds. The sigmoidal curve goes shwoop, like that. See? Chunk, 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 chunk. Because it's all broken up into chunks. See? I got answers. Awesome. We're into our cycles. This is where I really wanted to get, and we're doing okay on time. All right. Let's talk about these material cycles. So, is it all energy? No. There are other things that we need. Okay? We'll talk about minerals and water. Minerals, water, and carbon. You need those things. Like we already talked, remember we talked about biological macromolecules? And I was like, look, the amino acids and the nucleotides, they have nitrogen in them. You need to get nitrogen so your body can build those, right? Just that sort of concept. There's a really fun acronym. I said I wasn't going to get off on tangents. I'm going to do it real quick. Really fun acronym that they use in plant phys. SPONCH-MIG-CAC. Have you all heard about SPONCH-MIG-CAC? No? Nobody's told you about SPONCH-MIG-CAC?
no shot. That's a
It's a total, it's 100% is.
Phosphorus, oxygen, nitrogen, carbon, hydrogen, magnesium, calcium, potassium.
Exactly. Very well done. Points, you can have some John points. You get 2 of them. They expire at the end of the day, though. They're worth nothing. I'm so sorry. So this right here, these are sort of the primary plant nutrients, and that's how you, at least I was taught to memorize. And it's clearly it worked. I took that class like 15 years ago, 12 years ago. So there you go, SPONCH-MIG-CAC. We're only going to talk about potassium, carbon, and then water. So, yeah. Oh, and nitrogen. Cool. Magnesium, calcium, potassium. Yeah, exactly. And that does not sound like anything inappropriate at all, obviously. So I'll have you pull that right out of your mind. This is a college course. We take things very seriously here. Yes, this is like extended NPK. It's like NPK plus, right? Yeah. Cool. Great. Yeah, like realistically, if you're really getting into plant biology or plant phys, you want to know all of these, what the deficiency symptoms look like, why they're important for the plant, blah, blah, blah. But we're only going to worry about nitrogen, potassium, carbon, and water. All right, so water. Water moves through the system, right? The ecosystems. You've seen a diagram like this at some point in your life. I'm certain of it, okay? Water evaporates out of bodies of water. It precipitates out over the land. Plants need it to grow, and animals need it to survive. You got to have water. We're a water-based life, right? That's it. Water moves through in these cycles. So the water's, not really created or destroyed. It just sort of changes where it's held, and the salinity and the acidity of the body of water changes, and that sort of affects how accessible the water is. So we've got the hydrologic cycle where water evaporates. This is, of course, super simplified. Water evaporates up out of the big bodies of water and then precipitates over the land, driving the growth, driving the primary productivity, allowing terrestrial life to persist. Cool? All right, great. And you might be wondering, well, John, are we even going to talk about adiabatic lift at all? I know you're worried about it. Were you really? Oh. Well, don't worry, we will in week 5. I can't wait to talk to you about it. It's going to be fascinating because we'll talk about what makes the water precipitate out of the air. Anyways, cool. Carbon cycle. Ready? Carbon. We're talking about carbon fixation, where CO2 gets fixed into high energy organic molecules, and these high energy organic molecules get broken down. That energy is used, and the carbon is used to build other organic molecules. So you got a plant. The plant grows. The carbon came out of the atmosphere and the CO2, right? Then a bird eats a berry off of the plant. The carbon that the bird is going to be able to use to build up its feathers came from the berry that it ate. All right? Same carbon, same carbon, same carbon. You can track carbon flow through ecosystems using isotopes, which we talked about previously. Sometimes carbon gets sequestered for a very, very long time. All right? So carbon sequestration is an important concept. You want to control how much carbon is in the atmosphere, because those things act as greenhouse gases, those carbon-containing small organic molecules. Yeah? Probably not. No, I don't think so. No, I don't think so. Not beyond just talking about carbon sequestration, about the soil and the ocean being the major pools for carbon, right? We're not really gonna, yeah. But it's important, of course, you know? Yeah, awesome. So there's an entire, like I did my PhD research with biochar, and that's basically built entirely around making super stable carbon that then you put into the soil, you know? So anyways, carbon, all right? It comes out of the atmosphere. Sometimes after it gets fixed, it gets pushed into the soil or locked up in something like a shell, all right? And that makes it more stable, and that prevents it from being in the atmosphere. But then human activities, like burning of coal and fossil fuels, takes long sequestered carbon and puts it back into the atmosphere, and we end up with sort of an imbalance. That affects dynamics of how light energy gets captured on the planet, as well as other important things, all right? Adjustments to carbon dioxide oxygen ratios in the atmosphere, for example, adjust patterns of plant growth. Which types of plants are successful in which areas? Because some plants have evolved to deal with a low CO2 concentration in the atmosphere. If it goes up too high, those plants no longer have a selective advantage. They perish, and now the stability of the ecosystem goes down because you've got keystone species that are gone. Blah! The carbon cycle. Question?
So to be really simple here, the carbon turns into it goes into sugar, and then the sugar goes back into carbon dioxide.
Yeah. That's actually not bad. Let's go ahead and take a look at this right here. So, I mean, with plants and primary productivity, we always teach it like the plants carry out photosynthesis and they make sugar, but that's not true. Photosynthesis, the final output of photosynthesis is not sugar. It's this 3-carbon molecule that then goes off in branches into all these different pathways to build all the organic molecules the plant uses. So that's more accurate than it becomes just sugar. But then when we consume it, the glucose serves as a primary fuel, but you can eat things besides glucose and still get the carbon that you need. When you do cellular respiration, you breathe out carbon dioxide. It's the same carbon, right? Does that kind of make sense? I feel like I got jumbled. Kind of? Yeah. The carbon cycles, I have to learn this lesson. And people have been trying to tell it to me since I started teaching. It's like, sometimes just keep it simple and just breeze past it. I'm like, we can't though. We can't do that. Because if we just breeze past it, the students won't really understand. Then they'll think they understand, but they won't. And it's like, John, they'll learn it in an upper division course if they're interested. And I say, no, they'll learn it in my course. Because I think it's interesting. Like right now, you're going to learn everything about this. You're not. Yeah, you're not going to be tested on that. Cool. Cool. Good stuff. Well, actually, you are tested on the carbon sinks on the previous quiz, right? The carbon, so we were tested on the nitrogen cycle. Yeah. So here we go. Carbon, but always the quizzes will be answerable from the PowerPoints. the multiple choice questions, always. All right. Carbon dioxide, right? Let's do the basic overall view that you need to understand as a graduate of this course. Carbon dioxide is in the atmosphere. It gets fixed into larger molecules when plants grow. All right? Plants grow, they get consumed by other organisms, which then release some of the carbon, but hold some of the carbon. Sometimes carbon gets released quickly, sometimes it's sequestered and held for a while, all right? And how much carbon is in the atmosphere matters. Or in the ocean, because it's really easy to avoid marine ecosystems entirely, because I didn't study marine ecosystems, so I'm gonna talk about them as we move forward. Okay, nitrogen cycle. I'm gonna keep it so simple. It's gonna be so simple, don't even worry. Nitrogen. There is nitrogen in the atmosphere. It's the most abundant atom it's the most abundant molecule in the atmosphere. But within the atmosphere, nitrogen exists as N2 gas. And N2 gas is basically an inert gas that doesn't really do anything. We can't access it. We breathe it in, we breathe it out. So why can't we just get the nitrogen that's in the gas around us? We don't have the enzymes for it, okay? You need nitrogen. Nitrogen is in your amino acids that make your protein, and it's in the nucleotides that make up your DNA. Now, plants take up nitrogen in the environment, but not N2 gases, okay? Legumes have a symbiotic relationship with a bacterium that takes the N2 gas and fixes it into an available form. But that's exactly right, right? So, listen, getting at the N2 gases is difficult. Getting at the nitrogen is difficult. And the enzymes that nitrogen enters into the ecosystem in these ways. Dead material, it decays. All right. Excrement and urinary wastes introduce nitrogen. Lightning fixes some nitrogen and takes it from the gaseous form into a more accessible form. But the problem here is that once it gets converted into a moderately accessible form, it enters a soil nitrogen cycle where it gets converted between available and unavailable forms. It's all super complicated. But underlying it all are these bacteria with specific enzymes. So look at the Earth's atmosphere. It's nitrogen, oxygen, argon, and 0.04% other stuff. All right, cool. The Haber-Bosch process is the way that we make chemical fertilizers of nitrogen. You artificially generate nitrogen fertilizers. Then we've got nitrogen-fixing crops, okay, which really is what I want to get to. So, do we understand so far nitrogen is in the atmosphere, but it's unavailable for plants? Yes?
So out of curiosity, are like farts, would that be like N2?
I don't think so. I mean, you've all heard of course, like fecal blow-through. Yeah. So there's going to be some feces that's suspended in the gas. You know where the highest concentration of fecal blow-through is detected when they do swabs in the environment? Movie theaters. Isn't that great? Right on the seats. So just think about that. I saw the new Conjuring film on Sunday. We didn't enjoy it very much, but, we got to think about all the fecal blow-through.
What about college classrooms?
Yeah, I'm not going to think about that. I spend more time in college classrooms than in movie theaters, Yeah. Thanks for bringing up, farts. I appreciate it. Cool. Yeah, they are for cows, like that input of methane into the environment's, significant. So we got to talk about farts at some point. Anyways, let's look at this right here. What do I want you to know about the nitrogen cycle? The nitrogen cycle, okay, nitrogen's in all these unavailable forms, all right? But in the soil, it gets converted between available and unavailable forms by bacteria. There's a complex bacterial community involved in switching nitrogen between different forms. Yes?
Most plants can't get the nitrogen themselves, correct?
That's 100% right.
Through the bacteria?
Yes, so plants can form symbiotic relationships with bacteria that can get at the nitrogen themselves. Then there are also plants that form, there's a whole dynamic about that of some bacteria just associate with plant roots but never actually form nodules or anything. So getting access to nitrogen is incredibly important, but here's the cool thing, right? If you have a plant that can get at the nitrogen, when it grows, it has the nitrogen in it. And then when something eats it, that thing gets the nitrogen. Or when that thing decays, dies and decays, the nitrogen enters back into the system. So this is the sort of the principle behind doing things like crop rotations, where 1 year you grow a nitrogen fixing crop and you let it grow, and then you till all of the plant material back into the soil and let it decompose to get atmospheric nitrogen into the soil without having to apply chemical fertilizers, which can lead to their whole suite of other issues. Okay, so the nitrogen cycle's fascinating, legitimately very interesting stuff going on in the soil right here. Look at this diagram. I was really stoked to find this one. It's got enough detail, but it's not it's not excessive. There's some bacteria that are like denitrifying bacteria, and those are bad because they take readily available nitrates and they turn them into N2, and they get energy from doing that, and then they put it back into the atmosphere, and so they reduce available nitrogen in the soil. So if you're, say, interested in soils, you might be interested in how do we affect the microbial community so that denitrifying bacteria aren't a problem in the nitrogen cycle. You see how the more you understand about these things, the more nuanced questions you can ask when you're trying to carry out conservation, because conservation of soil microbial diversity is incredibly important for a a fully functioning ecosystem, right? But it's overlooked and avoided. Anyway, nitrogen fixing systems. There's way more detail than you need on this one. Plants make associations with bacteria. The bacteria can fix the nitrogen. Here's a process of nodule formation. Here's a picture of a plant with nodules on it. They grow out these little hairs, all right, that then wrap around bacteria, let the bacteria grow into their cells, and they make these little lumps on their roots, all right? And these are sites now of nitrogen fixation. It lets these plants dominate in areas that are nitrogen limited, and it makes them especially valuable if you're trying to, say, do a crop rotation or a green fertilizer, something like that. You want plants that can introduce nitrogen, bring it into the system, bring it into the cycle. Cool? There's a lot of other different ones. It's really fascinating. Some trees do it, birds do it, bees do it, educated fleas do it. Nodule forming. Let's form nodules. All right, nitrogen fixation. Red alder, ceanothus. You'll see ceanothus all around campus. It's a nitrogen fixer, okay? Red alder, it's a nitrogen fixer. There's a group of bacteria called rhizobia. Rhizo has to do with roots. Rhizobia is like the genus, and people are usually considering them as when they're talking about nitrogen fixing bacteria. But there's other species as well. There's even cyanobacteria that do it. It's pretty cool. Good. The phosphorus cycle, the last one, which will tie directly into mycorrhizae, and then we'll be done with the week 1 through 2 PowerPoints. Mistakes. Here we go. Phosphorus, commonly a limiting factor in the environment. So yeah, we're talking about phosphorus, not potassium. I forgot we were doing phosphorus in this class. It's my other class we talk about potassium. Okay, good. There's sort of phosphorus is often considered as sort of a limited resource in the soil because it breaks down and leaches out of rocks, but it then flows through the system pretty quickly. So phosphorus levels, if you're phosphorus limited, you can greatly affect plant growth and primary productivity by supplementing phosphorus, okay? So they're too water soluble is the problem. They're very soluble in the soil solution, so they can flow right out of the system. That's the big issue with these. So just like everything else in these cycles, you've got these producers that take the molecule up, and then they lock it up into a stable form, and then as they decompose, they release it in stable forms. All right? So the phosphorus cycle is slow. Phosphorus is a very common limiting factor, and getting the phosphorus out of the rocks is tricky. Okay? But one important helper that we can have in acquiring phosphorus are fungi. All right? They're very, very good at accessing phosphorus that otherwise would be locked up in rocks in the soil. Okay? So right here, real quick, phosphorus, just like everything else, limiting factor for plant growth. You need phosphorus in your body. One reason is for this molecule. This is ATP. It's that short-term energy currency. You need these phosphate groups to make it work. All right? Your body's making this all the time. You need phosphorus. Okay? Cool? I love it. Phosphorus cycle. Okay? It leaches out. Boom, boom, boom. It flows out of the system. Comes from mining, weathering. You got to mine phosphate rocks to get at phosphorus. It's a whole process of phosphorus mining for fertilizers. That's, complicated and da-da-da. But it's a very limiting factor. That's my main point. Okay, that's why we talked about it here. Mycorrhizae, all right? Fungi. What do we know about fungi? Well, they've got these little tiny... We're in ESM 105, and I was just talking to I just used the word hyphae and talking to some friends. That's what you call them. What are people who you hang out with but you don't get paid to hang out with them? Friends. That's what they are. And I said the word hyphae, and they were like, "What is that? Are you talking about E40?" And I said, "No. And you've just dated yourself as very old, just like me." All right. So here's the standard issue mushroom. Okay? This is a fruiting body of a mushroom. Underneath the fruiting body, you've got this complex connection, this complex network called the mycelium as a network. Okay? And the strands are called hyphae. Of hyphae. Here we go. So the fruiting body of a mushroom is a very small portion. It is also made up of hyphae, okay, that are sort of just compressed. There we go. Big network, it all grows together, all that stuff. And most of the time, the mushroom lives like this. No fruiting body. It's under the soil as a big network of hyphae. Okay? Now here's the cool stuff about the mycelium and the hyphae. They are very narrow, okay? And they carry out extracellular digestion. So Terry Hinkle will teach it sometimes as a mushroom is like a human turned inside out, where you're dumping all of your digestive enzymes onto the environment around you and then just absorbing the breakdown products over your membranes. It's pretty cool. So what does that mean? That means all of the hyphae here, the mycelium, right? They are exuding enzymes. These enzymes can break down very resistant molecules like phosphorus-containing rocks, okay? So the hyphae are very thin. They're thinner than plant roots. And they carry out extracellular digestion, so they help to break down the soil. They give you all sorts of other benefits, like increasing soil stability, detoxifying heavy metals because of this, okay? Enhancing nitrogen fixation, all sorts of really, really cool stuff about mycorrhizal symbiosis. What's mycorrhizal symbiosis? That's when the mycelium grows around or into the plant roots and effectively extends the plant's rooting system with a fungal network. It's very cool stuff. There are two broad types, endo, ecto. They're either on the outside or on the inside. Orchidaceous mycorrhizae are a specific type. The Brassicaceae family, okay, does not have mycorrhizae, and that's rare for an agricultural crop, okay? Really cool stuff. A lot of really cool stuff here. And then they can interconnect. So multiple plants can be linked together via a shared mycelial network, where then nutrients, water, other things can move between the plants, making the plants exist as more of a group, a single organism, rather than multiple separate organisms. Yes, the last question for the day.
Any high feed that's like not helpful to the plants or like harmful to the plants?
Oh yeah, tons. Yeah, no, there's pathogenic fungi that grow as hyphae in the soil. Like you'll see it in lawns all the time. Whenever you see like a ring-shaped rot, it's usually a hyphal network extending. And hey, I'll show this slide for 1 second because there might be some questions about it, and then we get into the week 3 and 4 material. It's going to be great. I'm so excited. Have a great day, everybody. Thank you for continuing to make it to class, and I will see you Wednesday.
Hey, thank you so much. Sorry, I wasn't in class on Friday.